All right, thanks for joining for Conversations with Faculty. We'll have Megan Corty up first to talk about the work in her lab. Thanks so much.
Okay, hi everyone. I'm going to share my screen. Hopefully this all works well. And
Looks
then
good to me.
just need to hit play. Play slideshow. Okay, so I am an assistant professor in the neuroscience department. So my lab is located in Gould Simpson. I'm going to tell you about the work that we do in my lab. And mainly we focus on one big question, or it's really two questions in one, which is how and why glia cells wrap axons. So let's break that down into what all of those terms mean. So in the nervous system, the fundamental unit of cell type is the neuron. And neurons are these highly specialized cells, and they have, I think you can't see my pointer, unfortunately. But you can see the cell body, and then they have these long processes termed axons. And the axons are what connect neurons to one another, so you can have information flowing in neural circuits to make your brain work, or information flowing along axons to reach down to your muscles to cause your muscles to contract so that they work. And these structures are these really long, thin structures, but they're super important. If they get broken or damaged or fall apart, then you can't transmit information anymore, right? And that's bad for the nervous system. And they can be super, super long. So in us, there's a neuron that lives at the base of our spinal cord, and we'll have to send one of these axons all the way down to the tip of your toe so you can wiggle your toe, or you can feel a pinprick in your toe, for example, right? So it's super long. And that gets even longer if you think about animals like in this example, like a whale or a giraffe, right? It's very, very long and very important, but also very kind of in a precarious position. And how does it survive the whole lifetime of an animal to keep functioning properly is one question we have. And it turns out that in the nervous system, neurons are not by themselves. And this is where my favorite cell type comes in. So there's this whole other class of cells in the nervous system called glia cells, some of which are diagrammed in cartoon form here. And maybe you've heard about them if you've taken a neuroscience class or even in a basic biology class. They're called astrocytes or microglia or oligodendrocytes or Schwann cells. They have all these names for all these types. But basically, they're these really important other category of cells that for a long, long time were totally ignored and just thought to be like glue holding things together. But now that we know they play a lot of important roles. And so now our challenge as biologists and neurobiologists is to figure out exactly what are the roles they're playing. Like we know they're important, but what are the molecules involved and the genes involved that allow them to do this? And so in my lab, I focus and study axon-related glia. So there's subtypes of glia cells that are just really associated only with axons. And this is across all different species in our central nervous system and our peripheral nervous system. And they have some common functions that we have understood some of them for a long time. So we know that they provide electrical insulation. So if an axon is like a wire transmitting electrical signals, if you insulate that wire, right, just like in normal, like in the walls in your house, then you're going to get more fidelity of transmission. And so we know that glia cells do this, but this is kind of a passive function, right? We know this speeds things up and makes things faster, which is great. But then in the last maybe 10, 20 years, we've started to understand that actually they're really important for keeping neurons and axons healthy over the long term. And we know that because in diseases like multiple sclerosis, for example, basically these cells that wrap axons die or get sick or fall apart. And we know that leads to neurodegeneration eventually and a lot of dysfunction for the patients. We also know that if we, like in animal models, knock out even just one gene from a glia cell, then the neurons can start dying, right? So how does that happen? How does changing a gene expression in a glia cell impact the neurons? And those are the kinds of questions we want to ask in my lab. We also know that these glia cells that are associated with the axons are really important for our responses to injury. So sometimes these glia cells can inhibit regeneration and fixing the injury, and sometimes they promote regeneration. And obviously, if we knew the molecules and the genes involved in that, we could try to bump up the regeneration and turn down the genes that cause no regeneration, right? Because those are not the outcome that we usually want to have. So these are the types of questions and the reasons why we study these cells. And I study them in Drosophila, and I do that because flies are small and easy to work with, and they have really quick life cycles, and we can get in and muck with all the genes of the fly really easily, so we can figure out the genes that are controlling all of these processes and all of these different cell types. And so this is the life cycle of a Drosophila on the one side, and we study nerves in both the larval system, so the kind of developing system, and also in the adults. If I took a larva, and I'm sorry that I don't have a, oh, here's my cursor. I don't know if you guys can see that or not, but I'm seeing it at least now. So in the third instar larva, if you cut it down the middle and open it up like a book, which is a dissection prep we do in the lab, this is what it looks like. The entire nervous system has GFP, this green fluorescent protein, so we can visualize it here. And these are the nerves that we study. And if I cut one in half and took it to an electron microscope, which is a really high-powered microscope that lets us see teeny, teeny, tiny details in cells. This is what the interior of one of those nerves of a fly larva looks like. So think how small that is. This is a 2-micron scale bar. So this is pretty tiny. And all of these individual kind of lighter colored things are axons, like coming right out of the plane of the screen at you. And then separating them is this darker membrane that I colored here blue, which is glia cells. And our nerves have things that look just like this. They're called Remac bundles. And this is an example from a mouse sciatic nerve that I also took an electron microscope picture of. And you can see that there are these round little axons surrounded by this, blue. It's not really blue in real life, but blue in these pictures, glia cells. And so this is what we study using the fly. So we can translate our findings to mice and humans, which is obviously humans eventually are what we're interested in. Okay, so we use the fly for a number of reasons. I told you that there's this morphological conservation, meaning those pictures look like one another. We also know there's molecular genetic conservation because we know that the the genes in the fly are really like the genes in humans, which is amazing, and it lets us do these experiments. And then we have these tools in the fly system for in vivo manipulation. What do I mean by that? By in a live animal. So basically, instead of having to take cells out and put them in a dish to change things about them or look at them, we can do this all in the living animal, which means that like those cells that are normally really close to one another, we don't have to rip them apart and then put them back together. And so it's really powerful to understand how these two cell types talk to one another, know each one another is there, respond to one another, right? And so some of the tools, which like I won't go into the details of, but basically they mean we can do multiple cell types at once. So I can single out the glia cells and I can turn on, for example, a fluorescent protein like this to make them glow red. And at the same time, I can turn genes on or off. And in the same exact animal, totally separately, I can do the same thing for neurons. So I can look at both cell types at the same type, which is pretty cool. And we can do all kinds of experiments using these tools. For example, like, do I want to prove the glia are important? Maybe if I just kill them and see how does that affect animal behavior. So I've done these experiments, and these are little Drosophila larvae that are normal, and they crawl around just fine. Hopefully you can see that on your screen. But if I kill off the glia with genetic tools, you can see like this little guy is kind of moving, and this little guy is kind of moving, but really they're not doing so hot, right? And so we have computer programs that let us quantify their behavior so we know like how badly they're affected when we do certain genetic manipulations, maybe not as strong always as killing the cells entirely. So we can do things like single out individual axons with, again, a bunch of genetic tricks. So this is one axon from one neuron, and I'm watching it move little vesicles back and forth, which I know is a sign of whether the axon is healthy or not healthy. So that's how we can monitor axon health. I can see if the neurons respond to stimuli. So here are neurons that are responsive to sugar. So you'll see thing glucose comes on, and then boom, when they sense the sugar, you can tell that they were actively responding. So these are the types of experiments that we do in lab. We can also prod these flies, kind of tickle them with an eyelash to see whether or not their sensory capacity is then. So you could tell that he was kicking a little bit because he could feel something on him. If we damage the nerves in some ways, they don't know this, and you can keep poking them and they don't realize something's poking them and they'll never kick it away, right? And these are the types of experiments we can do in the lab. And so I said we answered two big questions. So basically in my previous work as a postdoc in a lab, I did two big genetic screens to identify new genes that control both the two questions I asked for. How do these cells develop? And then how do they support axons? So the basic premise is like this with flies. I just do a single cross of flies, and then I can get progeny where I can look at the glia and I can knock down genes one at a time and just ask like, when do the glia start looking weird? Basically was the premise of this. And I did it 2,300 times to test 2,300 genes, dissected those animals, imaged them on a fluorescent microscope. My lab has its own confocal fluorescent microscope for students to use. And then look for glia that look weird. And when I did that, I found about 100 times that the glia looked weird. And so those are all genes we have to follow up on in my lab now with students to see exactly how does that gene work to make a glia develop properly. Eventually, we look at those by things with like electron microscopy, so we can see those individual axons and how they're wrapped or not wrapped, or if they look sick or healthy, and quantify that. So tracing axons and doing a lot of image analysis is a big part of the lab. And then for the second question, we also have a set of genes that answer that second question. How do glia support axons long term? And so again, with just like kind of proving whether or not this experiment would work, I went ahead and killed off all the glia completely. And I saw that in control cells where I look at this nerve and I see all these little axons running by these green lines, they look pretty good. But if I've killed the glia, then they start looking kind of cruddy, right? You can see that there's thinner, so there's fewer neurons there, and they're blebbing and they're leaving debris behind. It's not great. So then I asked the question, if instead of killing the entire glia off, what if I just took out one gene at a time over and over and see if any of them make these neurons look sick and unhealthy? And again, I did that about 2,000 times and found 70 times where the neurons look sick. And so these are genes that we'll follow up in my lab to say, okay, how does changing gene expression in a glia cell actually impact the health of a neuron? And so following up on this is what we will be doing in my lab. The experiments are a lot of fly genetics. So we can look at flies under the microscope and see all these markers they have. So they have different color eyes, different shaped wings, different shaped bristles. You'll get to learn all of those if you join the lab, and then select the right ones to set up in crosses. And then you do, kind of back to when you learned about Mendel and P's and stuff, these Punnett squares to figure out what are the animals you're going to select to use in your experiments in the end from the cross you did. And then dissect them, use antibody staining to image them under the microscope. Sometimes do live imaging where we just cut the wing right off the fly like this with a little pair of scissors, or we can do an injury by just cutting the tip of the wing off. And then looking at the fly and the wing stub that it has left to see how those sick axons are responding, if the glia can help them get better or not. And then, of course, we do a lot of molecular biology. So PCR, cloning, Western blots, that type of stuff. So those are the techniques you'll learn if you join the lab. Those are the kinds of questions we ask. So if you're interested, oh, I have some more pictures. But if you're interested in joining the lab, please get in touch with me. Here's my email address. And I'd love to meet with you. I'm looking to get a couple students in the lab for the summer. Again, we're in Gould Simpson, and I will take any questions you guys have about what the lab does. Happy to answer questions. I don't know if I can see everyone, if anyone's raising their hand.
Questions? Yeah, we have a few minutes for questions if anybody has them.
Okay, I have a question.
Sure.
I was wondering like what exactly the size of your lab is, like how many people do you have working in it, and what's like the
That's a great question. The
Okay.
current lab has 0 people. I arrived last month. And so right now it's just me. So for the summer, it will be a technician that I'm looking at hiring right now, and then probably summer undergraduates, and then towards the end of the summer, rotation grad students. But the lab is quite small right now because I've just begun. But that means everything is new and shiny also. So lots of projects to be had.
Cool.
And then I have a question as well.
Sure.
I know you mentioned looking at those 100 genes that you identified. I guess once you work through those 100 genes, are you looking more at developing the pathways for development, or are you looking to maybe look at human disease research?
Yeah, so basically these are the genes that are the jumping off point. So each one will probably spawn years and years and years of research in the end as we work out like little bits. And so the idea is to look at everything. So depending on what people's interests are in the lab, people can pick hits that are from the developmental screen, or people can pick hits that are from the functional screen. Although I will say there's a lot of overlap. So in the genes that I've worked up so far that had developmental problems, obviously then they don't wrap normally. And we realized that actually that contributes to neurodegeneration in the end too. So a lot of these things are kind of hand in hand. We have some examples where they cause neurodegenerative phenotypes, but they don't have any developmental problems. And those are interesting because like they look normal. So then what's the problem? The ones that cause developmental phenotypes, like maybe the problem is like nothing developed right in the beginning. So figuring out why that is and why that leads to the neurons dying as the animals age is important to figure out. So it's a little bit of everything. Luckily right now, there's enough things for people to choose from that you can choose kind of your own adventure in terms of what types of experiments are done in the lab. But luckily we have a lot of stuff to follow up on.
Thank you.
Mm-hmm.
We still have a couple more minutes for questions if anybody has them, but if not, we can move on to Dr. Gallery.
Right. Thanks for coming.
Megan, I'll ask a quick question.
Yeah.
I'm your neighbor in ACBS, so I'm like across from you. How fun is it to work with Drosophila?
Drosophila is super fun. I like working with the flies a lot. I think it's really cool. I don't know if anyone here has worked in other animals before. I've worked with the Drosophila for a long time. And one of the fun things about them is we don't have to genotype them. So if you've ever worked in a mouse lab or any other lab, to figure out, after you do a cross and you're looking at like the progeny, the babies, you figure out which ones are the right genotype, like which ones have the mutants or which ones do this. You have to like very tediously do PCR. No shade to any of the people that do mouse work. I've done this a little bit and it drove me wild because with the flies, you literally just look at them on the pad and by their eye color and their wing shape and their bristle pattern, you automatically know which one is the right one. And so it saves a lot of time to do more of the fun stuff and less of like the tedious stuff behind the scenes. And yeah, they don't bite you. This was also a criteria I had when I started in grad school. I was like, oh, I want to work with animals that don't bite. So I worked with fish and flies, neither of which can bite you. So if you're worried about that, which I was very worried about getting bit by a mouse, then a fly lab might be the place for you.
Awesome. Thank you so much, Dr. Corty. And
Thanks.
yeah, we'll move on to Dr. Gallery. Thanks so much for being here.
Well, first I'll say welcome, Megan. Welcome to the U of A. How exciting to set up your lab. That would be a wonderful opportunity to, I'm just saying this to the students, to get in with new PIs because they have lots of resources, lots of ideas. So, hello, everyone. I'm Rachel Gallery. I'm sick, quite sick, actually. So sorry. I'm a professor in the School of Natural Resources and the Environment, professor of microbial ecology, and I'm also the Thomas Lovejoy Endowed Chair and Director of the Center for Bridging Biodiversity, Conservation Science, and Policy, which is a fairly new initiative that I'm really excited to get going. What I'm here to talk with you all about today is a really national project that we've been working on for many years through the Critical Zone Observatory Network. This is a National Science Foundation project, and we have a site here in Arizona that we'll go into a little bit more detail about. So first, I just want to set the stage here and say that probably many of you do not think about soils a lot, and that's okay, but soils are important. Yeah, first of all, soils are a non-renewable natural resource, meaning that they cannot be produced during a human lifetime. You know, it takes thousands of years to form just a few inches of topsoil that can be destroyed through poor management in just one growing season. And soils are literally the foundation of society. You know, Teddy Roosevelt said, a nation that destroys its soils destroys itself. But how do soils actually form? Well, you know, that is the question that you as a UBRP student would be working on with us over the summer, trying to figure out how soils form across all of the different types of ecosystems that we have in the US. So as I said, this is a critical zone network. And what that means is that we have multiple sites where we have the same types of experiments going on. So down here in Arizona, we have a research site up on Mount Lemmon at the Bigelow Research Tower. We also have sites out in the Biosphere Desert. But we've also been able to collect soils from around the US, from Puerto Rico, from, let's see, the grasslands of Kansas, from over in the Sierra Nevadas, way up in northern Idaho, and a couple of other places that I have forgotten. And all of those soils are now back here in our research lab. And what we would be looking for help with this summer is for students. Well, I think you would have seen the blurb already, so I'm not going to read the whole blurb. But basically, we have a bunch of really technically difficult but interesting lab work to do on these soils to understand how the microorganisms, so the bacteria, the fungi, the archaea, possibly even the viruses that are living within these soils, how their respective metabolisms are helping to weather and eventually form soils. And we have a series of hypotheses about how this might be occurring differently depending on the underlying bedrock or the type of parent material that these soils would be forming on. This is going to be a project that's run by, over the summer, you're going to be primarily mentored by the third-year PhD student, Alexander Ederer, who is in charge of this research project. But you'd be working across a team of folks from my research group, and I'm a co-PI, or I'm the lead PI of this project, and our co-PI is John Chorover, who's a professor in the Department of Environmental Sciences. I have an 8-minute video that I was just going to play so that I don't talk, because Arizona Public Media just did a piece on the critical zone. And so I think it might just be more fun for y'all to listen to this, and then I'll take questions if that sounds okay to everybody. Let me share my screen. Go here. Here we are. Is it this one? I think it's this one. think, is that right? Everyone can see that video?
Looks good. Yeah.
Okay, here we go. So like I said, Arizona Public Media just did a little piece on this, 8 minutes, let's go.
I have felt most relaxed, most at peace, and most also stimulated, and I guess happiest when I've been out in the natural world. And I regain that feeling out into the critical zone observatory sites. Here in Tucson, we live at the foot of the Catalina Mountains. And as you drive up, you go through a variety of ecosystems. You go from the Sonoran Desert ecosystem down at the foot of the mountains through savanna woodland. You keep going a bit higher and you start to see ponderosa pine trees. And you go even further and you get into what's called mixed conifer forest. The Critical Zone Observatory was designed to take advantage of this gradient. Chilly.
Hi, I'm Nicole Hockley.
Oh, man. Give me a
I
sec.
know this is hard to hear, but it's important. What is critical zone science? Critical zone science requires understanding the physical, the chemical, and the biological factors that influence life on Earth. So you need to know all of it. It encompasses everything from where groundwater interfaces with the bedrock, all the way up to where you have atmospheric exchange occurring at the tops of tree canopies.
We actually live on and take advantage of a very thin layer of the Earth's surface. And it's termed critical because arguably all of life on the planet depends on the presence and functioning of that portion of the Earth's land surface.
The critical zone, in a variety of ways, spans across an enormous multitude of scales, temporally and spatially. We can think about an entire hill slope or even an entire basin, all the way down to a tiny nanoparticle. And part of my work is aiming at looking at the smallest constituents of the critical zone and contextualizing them within larger scale processes.
So when the Critical Zone Observatory program was first developed, the National Science Foundation conceived it as a network of sites where all of the sites would have specific science questions they were trying to answer. And we were selected here in Arizona in part to represent the hotter and drier end of the climate regime. What we want to do is to understand what are the feedbacks between the microbiology and the hydrology and geochemistry, and how do they couple with each other and interact? And you can only do that by bringing the data sets together as a team and looking at the cross relationships between them.
The different bacterial and fungal communities that exist up at the surface are actually going to be fundamentally different, both in terms of who they are and the type of work that they do as we go deeper down into the soils. So we often think about, how are soils formed? It's through the physical and the chemical, but also the biological weathering of rock. And so one of the current research questions that we're really excited about and really interested in trying to answer is, how does the interplay between what's happening at the surface and then what's happening in the subsurface, how does the interplay between those processes create the evolution of soils and influence the distribution of the microbes that are living in and along this entire soil profile?
Using this vacuum pump, I'm able to extract water from the soil into this collection bottle, which we can then bring back to the lab and analyze. Soil is fascinating because it's where the non-living meets the living. And here in the Santa Catalina Mountains and the Critical Zone Observatory, we're able to look and see how these soils differ from one another and how we can relate those to environmental variables like climate and like the parent material, the geology of the place. The geologic underpinnings of the critical zone took hundreds of millions of years to develop and are forming into what we currently observe today. And at the same time, a lot of the chemical reactions that take place in soils can happen in under 15 minutes.
We also have folks like Craig Rasmussen who have been looking at how this process of water movement through this catchment is transforming rock into soil.
We're trying to understand the long-term evolution of this whole landscape. So behind me, there's a great example of one of the natural processes here that moves and redistributes mass and material across the landscape. We have trees, as they fall, you can see it brings up a big root ball with it, which brings a lot of soil. That soil can then move by gravity down slope. What I enjoy is going back to the same landscape repeatedly. You see the landscape as a whole and how it all fits together, and you're like, oh, I can see how this whole process works. Soils are kind of it's the story, and it's not always easy to understand the story, so you have to decipher it and translate that from what you measure to what's going on in the environment.
I think that the critical zone and studies of the critical zone are essential for being able to not just predict the future of the Earth's surface and the world that we live in as humans on the planet into the future, but also to be able to develop solutions to many of the problems that we've created for ourselves.
The real value that I find in critical zone science and having this perspective and why it's so exciting is because we are facing so many different types of environmental challenges or even environmental crises. We are experiencing rapid climate change. We are experiencing more frequent droughts and hotter temperatures here in the Southwest. And if we don't have a holistic understanding of a system, then we are not going to be able to make meaningful and informed decisions about how we might manage that system. We have to remind ourselves that as humans, we are part of the ecology of this biosphere. We are part of the organisms that are interacting with the other organisms in this system. And so we have an inherent role there as well.
My hope is that through learning about how humans have an impact on critical zone processes, we can move our interventions and our approaches to critical zone management to a place that will sustain the health of the ecosystems, that will allow us to be nourished by those ecosystems and be sustained by those ecosystems long into the future.
Okay. All right, well, thank you for listening to that. I think it did a better job than I could have just now in talking about our science. So I'll just add that in terms of practical things that you'd be doing with our group this summer, that would include some field work in those sites, but also it would include, as I mentioned, lab work with a bunch of the soils that we've already collected. And again, you'd work very closely with Alexander Ederer. But then the other element is that because this is a critical zone network, we do have all hands meetings with the larger project team, and students would be invited to join and participate in those so that you could get a greater sort of sense of the bigger project. And then finally, we do have an in-person all hands meeting scheduled for the end of this summer, and we pay for travel for particular students and faculty to attend those. And if a student working with our group had a poster presentation that they could present, we'd be really happy to pay for all of the travel and accommodations and to provide that professional development experience. So happy to take questions.
Any questions from the group?
Not a bunch of ecosystem ecologists here. That's all right.
I'll ask a question. No one else is. This is fascinating. For working with the soil, and I don't know if I just missed this in the beginning of your talk, does your lab focus on working more looking at soil components and what it's made of, or the microorganisms like you were talking about, and figuring out what the different types of bacteria or fungi are at the different levels? Is it or all of the above, maybe? I don't know.
Yeah, so we do both. The project title is actually Biogeochemistry and Microbiology of the Critical Zone. And so I wasn't talking about an entire element of our project that involves all of the genomic sequencing of the elements of the soil. So all of the, bacteria, fungi, archaea. We have that entire project going on. Essentially, every soil sample that we collect, we're getting an entire picture about it. So we're trying to get the full biogeochemical profile plus the microbiology. And that, of course, helps us to understand the relative importance of the physical, chemical, and biological properties that are helping to form those soils. And so I wasn't really mentioning the microbial sequencing part so much because the student this summer would be working more on the geochemical side. However, if our student has an interest in knowing more about that, the microbial, we call it the metagenome. We use a DNA-based sequencing. We're working on the RNA stuff, but RNA in the field is such a nightmare. But yeah, thanks for asking that question.
Cool. Yeah. Maybe I'll get a summer internship.
Yeah, me and yours too. Maybe we can.
Thank you so much, Dr. Gallery, especially for being here while you're sick. And we are recording this, so we're going to post it. So all of our students who are looking for labs will see your video and stuff. So yeah.
Wonderful. Okay, well, thank you so much.
Yeah, of course. And then, now on to Dr. Duca.
Hi, yeah, thanks. So thanks for the invitation to present. I will share my screen. Okay, cool. Okay. Yeah, so my lab is also located on the main campus, actually really close to the Simpson Gold Lab, across from 6th Street Garage. And we've had several UBRP students in the past, and they've all done amazing jobs. And so we would love to have more UBRP participation in our lab. And all of my graduate students get very excited when they can work with a UBRP student, because they know that they're going to be committed and really excited about what they're doing. So my lab, as you can see, has gut feelings about obesity and diabetes, and we're really interested in the role of the gastrointestinal tract in the development of metabolic disease. So Hippocrates is coined with saying the phrase, "all disease begins in the gut." And this quote is actually on our door in our lab. And we actually feel really strongly that a lot of diseases are due to a dysregulation in the gut and with the microbiome or the bacteria in the gut, which I'll get into in a little bit. So this is a little animation here that's going from 2004 to 2019, and it's showing the increase in obesity and diabetes prevalence. So as the boxes get more blue and more red, and then eventually more purple, which is a combination, it's showing that there's an increase in the rate of obesity and diabetes. So if I just play that again, you can see, not gonna lie, 2004, the country looks pretty decent, but by 2019, you can see this huge influx in the amount of obesity and diabetes in our country, where now most states have rates of over 30% of their individuals being obese and over 13% being diagnosed with type 2 diabetes. So a huge, huge amount of individuals that desperately need help to alleviate these diseases. So in a very, very simple sense, obesity is an imbalance or tipping in the scale, where normally there's a body weight maintenance where the amount of energy you take in is compensated by the amount of energy you expend, or energy expenditure. But for body weight gain, there's an increase in the energy intake compared to energy expenditure. And our lab is really interested in this energy intake side. So what is causing individuals to overconsume during a meal and overeat the amount of calories? And there's a lot of evidence that the gastrointestinal tract, or the gut, contributes to how much you eat on a daily basis. And this is known as the gut to brain axis. So in a very simple sense, you ingest nutrients during your meal. So this is your carbohydrates, lipids, proteins. You ingest them in during a meal. The first thing that they pass through is your GI tract, where then they can be absorbed into the blood and go to all your peripheral tissues as energy sources. So before they're even absorbed, there's these really specialized cells called enteroendocrine cells. So here's one in blue, that they can sense those nutrients. So they're not involved in the absorption, but just sensing that you have a meal in your gastrointestinal tract. You're about to absorb a ton of nutrients. And what they do is they sense that, they become activated, and they release these peptides or signaling molecules. And those signaling molecules will activate neurons that innervate from your gut all the way to your brain. So Megan gave a very good presentation of how long some of these neurons can be. So there are neurons that are coming from the hindbrain, from your hindbrain, and innervating all the way down to different parts of your GI tract. So nutrients activate these cells to release these peptides, to activate these neurons that signal to your brain to tell your brain, hey, you're consuming this amount of carbohydrates, this amount of protein, this amount of lipids. And what you can start doing is to decrease how much you're eating because you have enough to eat. And in a very similar sense, we've kind of shown that it also signals to your brain to stop producing glucose. So your body has a very maintained level of blood glucose, and your body is actually producing its own glucose to maintain those levels. But when you eat a meal, you have a lot of glucose coming in. So this is another way that your gut can tell your brain to stop producing your own glucose. You don't need it because you're about to get a lot of glucose. And that prevents hyperglycemia, which is a chronic condition of type 2 diabetes. So our lab is really interested in basically everything on this slide. So how do nutrients activate these cells? How do the cells release these peptides? How do the peptides activate these neurons? And how do they signal to the brain? What are they signaling at the level of the brain? And how ultimately does it decrease food intake and glucose production? And we're really interested in all that bacteria that is in the gut that can be influencing this pathway. So the bacteria can interact with these nutrients to kind of create like metabolic byproducts, so slightly different nutrients than you consume. They can actually influence these cells. They can influence the neurons themselves. So a whole lot of effects on how the microbiome can impact that gut to brain signaling. So to do that, we use a lot of in vivo techniques. We're a physiology first lab, so everything we try and do in vivo to make it as physiologically relevant as we can. I'd asked Megan about the flies because I've never worked with flies, and all I've worked with is mice and rats. And I can actually say I've been working with them for 20 years, and I've only been bit once in my entire life. So not so bad as long as you have, I guess, good teachers. And I think our lab are really good students and really good teachers. But we do a lot of really interesting surgical techniques that we can do in our mice and rats to kind of get at this gut to brain signaling. So we use a lot of obese and diabetic rats and mice. When I say obese, there are rats that are about 2 pounds, like 1 kilo that we have. So really big rats, and they kind of just look like big fluffy softballs with little heads. They're actually kind of funny looking. And we also use what's known as germ-free mice. So these are animals that have no bacteria, no microbes at all. So we're allowed to be able to study how can certain bacteria impact physiology by using mice that have no bacteria or introducing specific bacteria. And that's kind of what this looks like down here. This is a germ-free isolator. So it's kind of like if anyone is a fan of Seinfeld with the bubble boy, it's kind of the same concept, except they're like bubble mice. And they're legitimately all no microbes within that bubble. But we do a lot of surgical techniques. So we do things like intestinal catheters, where we put a catheter directly into the intestine of the mouse or the rat, and then they are completely normal. They just have a little tube coming out the back of their neck, and we can infuse whatever we want into that catheter. We can do the same in the jugular vein, the carotid artery. We can do gastric bypass. We can do a vagotomy, which is cutting those neuronal connections. We can do actually really neat things now where we can actually inject specific viruses to target specific proteins within those neurons. So this is a cool surgery we just learned a few months ago. So I'll just kind of want to show this video. So this is what's called a no-dose ganglia. So it contains all the cell bodies for the vagus nerve, which is that nerve that runs from the gut to the brain. So if you want to target specific proteins on those neurons, you need to inject viruses directly into the cell bodies. So it's basically like a little clump of thousands of cell bodies. It's about the size of pretty much like a grain of rice. And what we can do is we can have a little micropipette here. This is all done under a microscope, and we can really, really finely under that microscope, get that micropipette and inject and infect those cell bodies right in that no-dose ganglion. And again, that's about the size of a grain of rice that my student is injecting here. So some of the cool in vivo surgeries we can do. And again, what we're using this for is to test in vivo how the microbiome can impact that gut-brain axis to regulate food intake, regulate glucose levels to ultimately contribute to diabetes and obesity. And I'll just give one little example here. So we do a lot with what's called prebiotics. So these are mostly like non-digestible carbohydrates or dietary fiber that you can consume that have a beneficial impact on your health. And the main there's many that people kind of take or use, but it's not really known which ones are best for weight loss and improvements in glucose homeostasis. So we did a study where we took mice and we put them on a high-fat diet to make them obese, and then we supplemented that diet with a bunch of different dietary fibers. So we did beta-pectin, so pectin's really high in apples, beta-glucan, that's high in barley and oat flour, wheat dextrin obviously is going to be in wheat flour, oligofructose is high in things like chicory root and Jerusalem artichoke, and resistant starch is a kind of a newer dietary fiber that people are putting into foods. And what we found was only 2 of those fibers, the oligofructose and the beta-glucan, were able to kind of reduce the amount of fat that those mice developed over time. So we think these are potential beneficial dietary fibers that people can use to supplement in their diet for weight loss. And it can also improve glucose homeostasis. And that kind of led us to, how is this happening? So how are these fibers improving weight loss? So just to give a real quick example of like an in vivo study that we do, we have a rat, and basically put this rat on a high-fat diet to make it obese. And then we put a cannula directly into their small intestine. And again, this cannula just comes out the back of the neck. The mouse or the rat doesn't even know it's there. And that allows us now, whenever we want, to hook this up to a pump, and then we can infuse a nutrient directly into their intestine and see how their intestine can sense that nutrient and signal to the brain to lower food intake. So to do that study, we fast the animal overnight so they want to eat, and then we infuse either a nutrient, so in this case it's a lipid, or a saline, which is a control, and then we give them their food back and we record their food intake for 2 hours. So in a normally healthy chow-fed rat, so this is a rat that is on a healthy diet, they're not obese, compared to that saline or control infusion, when we infuse a lipid into the intestine, they sense that lipid, they signal to the brain, hey, you have nutrients in your intestine that are about to be absorbed, you don't need to eat food. So what do? They decrease their food intake about 80%. So they don't eat because they have lipids and nutrients in the gut. However, on the animals on a high-fat diet, they don't decrease their food intake. So what that means is that gut-to-brain signal is lost. So during a meal, they're overeating. And that constant overeating on a daily basis leads to obesity and weight gain. So what we did is we took those animals and we switched them to the same high-fat diet. So they're still on a diet that's super high in fat and sugar, except we added that prebiotic oligofructose and we retest them. And we see it completely restores that gut-to-brain signaling. So now they don't eat as much during a meal, and that's why we think they're losing weight when we supplement with that oligofructose, is it's restoring that gut-to-brain signal. And we do a lot of other things, like we can look at the amount of peptides that are secreted. We can look at the activation in the brain and see if that activation truly is increased. And another really neat thing we can do is we can actually swap their microbiomes. So we can take the bacteria from one animal and give it to the other animal and see if that's what's causing this effect. And what we do see is that is what's driving our effect, is that it's the diet altering the bacteria in your gut. And then when we give just that bacteria in the gut and not the actual oligofructose diet, we can still restore that gut to brain signaling. So there's a lot of interactions going on in the gut and in the diet. And that's kind of what we're teasing apart using our in vivo surgical techniques and surgical models. So with that, I'll stop. And this is our lab. This is pretty much the current lab we have now. And this is our lab mascot. This is my dog Beans. You can see here. He comes to all the events. So you'll be able to hang out with Beans every once in a while as well. So with that, I can take questions if anyone has any.
Any questions from the group? Arpita, do you have a question or no?
Yeah, I guess I was kind of wondering, where did you pick the prebiotics that you chose? Are those things that people commonly use, or are those things that people don't use as often, or what's the inspiration behind that?
Yeah, so that was a grant we had from the U.S. Department of Agriculture. So those are all plant-based fibers. And the goal was that we want to see what plant-based fibers were most beneficial, so we could inform the agricultural community. These are the fibers that, you know, individuals in America should be eating on a more regular basis, so to increase the crop production of those specific fibers. And then on the other side, to be able to tell people who, you know, need to lose weight that these are the fibers that you probably should be consuming more. So it was kind of, it was more like we chose plant-based fibers because it was from the USDA.
Thank you.
Mm-hmm.
Anybody else? Oh, yeah. Oh, oh no, that's Dr. Gallery. Okay, well,
Awesome.
with that, thank you so much, Dr. Duca.
Yeah,
Also,
thank you.
Tell Beth hello. I used to work with her in the micro labs. We
Oh,
taught
awesome.
421 together at the same time. So yeah,
That's awesome.
I was
I
happy
will.
to see her face. And thank you so much, Drs. Corty, Gallery, and Duca, for being here. And then to the students, we'll have another conversations with faculty on Friday at noon. So come to that or look at the recording online. And yeah, thanks again for being here, everybody.
Thanks
Thank
for
you. See ya.
organizing.
Thank you.
Thank you.